I am honored to introduce our speaker for today, Dr. LaTasha Crawford. Dr. Crawford obtained her bachelor's in molecular biophysics and biochemistry at Yale University. She then earned her veterinary medical doctorate and PhD in neuroscience at the University of Pennsylvania. She went on to complete her residency in veterinary anatomic pathology at Johns Hopkins University and stayed on there as a postdoc before landing her current position as an assistant professor in the Department of Biological Sciences at the University of Wisconsin School of Veterinary Medicine. Dr. Crawford's research investigates mechanisms of pain with a focus on sensory innervation of the skin and bladder in mouse models of disease. She is particularly interested in how sensory neuronal subpopulations play distinct roles in nerve injury, pain, and in bladder pain. Her talk today will focus on referred pain in bladder disease and whether referred pain could be about more than just a confused central nervous system, which sounds pretty cool. So please do tell us all about it, LaTasha.
Well, I will be presenting today on some of the work that my lab is doing and the projects that we are working on. In general, my lab studies pain, and what we've been doing is actually approaching bladder pain with the lens of what we've come to understand about nerve injury pain and things that happen primarily to primary sensory afferents after nerve injury, and kind of taking a lot of that information and using that as the lens through which we actually approach bladder pain and bladder dysfunction. And so one thing that I thought I would kind of start out with here is a little bit different than some of my other talks, but what I thought I would start with is just this idea of referred pain. So it's something that we've all kind of heard of. It's something that we've all thought a little bit about. A very kind of typical manifestation is for folks that may be having a heart attack. So their heart is undergoing some sort of pathology or ischemic damage, and you feel that pain in your arm. And so we've all learned from textbooks that this is really a result of neurons that are in your spinal cord that are receiving input from, say, the skin of your arm, in addition to some part of your internal organs, like your heart in this example. And that spinal cord neuron can't discern where that input is coming from. And so this is a little bit of a diagram that kind of illustrates that I thought was a really common understanding that we often have. And so one of the questions that we have, though, is what if this is not just a mistake that's made by the spinal cord? What if there's someplace else where sensory neurons coming from these two very distant spots may actually interact with each other? And so our focus has actually been on the dorsal root ganglia, shown here. So through our studies in understanding more about pain neurobiology after pain and neuropathic pain, so after nerve injury, we've come to have a really thorough appreciation of the dorsal root ganglia and some of the emerging understanding that we have of sensory neurons that are housed there. So this is an image of a mouse dorsal root ganglion. There are many different types of sensory neurons that are housed therein. These neurons vary in terms of the cell body size. In this case, we've done labeling with different immunomarkers, and there are also some genetic labels to indicate different subtypes of neurons. But I think you get the point that there are lots of different types of sensory neurons there. So it's a very heterogeneous population. They differ in terms of their neurochemical markers, ion channels they may express. They differ in their ability to interact with the immune system, different receptors for immune molecules and things of that nature. They are the coolest part of the neuron of the dorsal root ganglion, I would say, as a person who's obviously a little biased. But there are other cell types that are there as well. And these are really cell types that have become more and more important for pain. The more and more we understand about them, the more and more we understand their roles and their importance for that. So there are not just neurons here. So the neurons are kind of the larger cells that we're seeing here. They communicate a little bit differently than what we see in the central nervous system, using both glutamate and GABA. They also communicate with satellite glial cells. So a lot of these kind of denser, darker cells with condensed nuclei, these are satellite glia that are surrounding these particular, these neurons. And I'll show another image of that in a moment. They can communicate via gap junctions with those cells. And there are also resident macrophages that I'll show in a moment as well that are always present within the dorsal root ganglia, and that can also modulate communication. Another thing that's unique about the dorsal root ganglia is that there's no blood barrier. So unlike the central nervous system, there is no blood-brain barrier in the dorsal root ganglia. And in fact, the endothelium is lined by the blood vessels are lined by fenestrated endothelium, which means that dorsal root ganglion neurons have ongoing exposure to circulating metabolites, signaling molecules, cytokines, drugs, if it's a patient that's, undergoing therapy. So all of those things can reach the neurons, sensory neurons, very, very easily, probably more so than a lot of other tissues in the body, which is really interesting. And so there really is a very unique type of intercellular communication that goes on within the dorsal root ganglion. Here is, actually I'll show on the next slide, it's a little bit better. So these are just some immunomarker images that can demonstrate those satellite glia as they surround the neuron cell bodies. And you can see that sometimes those neuron cell bodies are actually somewhat clustered and are surrounded by satellite glia that themselves can actually interact with those neurons. And in pathologic conditions, we can actually see neurons start to communicate with other neurons that are within those clusters. The neuronal markers are shown here in kind of red and blue, just to give you a sense of how these satellite glia surround them. And then I'm also showing here that there are not just satellite glia shown in kind of the magenta color, but also macrophages that are here all the time and that are actually increased in number after certain types of pain scenarios or certain models of pain. And so these are all kind of part of this milieu, this environment in which all of these cells are sitting. All of these neurons happen to sit there and they can communicate with each other. And how that communicate changes is something that we really are really just starting to understand, understanding it mostly in our mouse models, but the way in which we understand how the dorsal root ganglia change in patient populations is actually really poorly understood. Because it's not something that we look at very often. So I alluded to all of these different types of neurons that are here, and I won't go into a ton of specifics, but suffice it to say, there are a lot. What we know about sensory neurons, we know the most detail about sensory neurons that innervate the skin. And there are over 15 different subtypes that all have very unique patterns in terms of their morphology, what they actually innervate in the skin, where they send their axons to within the central nervous system. And you can get a sense of that just by kind of the color coding that's kind of at the bottom here. But suffice it to say, there are many of different types of neurons. They have distinct roles and different types of sensation, and probably as distinct susceptibility to injury. And we think that that also means that they have a distinct plasticity or distinct potential roles in pain or in disease. So in our approach to really diving into bladder pain, we really want to look at cell-specific mechanisms. So we're very interested in what's going on in the dorsal root ganglia and the way that sensory neurons are communicating with each other, but also looking very specifically at different subtypes of sensory neurons as we're diving into these mechanisms. So ultimately, there is another place where sensory neurons can communicate, where sensory neurons that innervate the bladder also communicate with sensory neurons that innervate the skin, if we're looking particularly at bladder pain. So this dorsal root ganglia is pretty key. And there's another very interesting thing that has emerged from our understanding of pain mechanisms after nerve injury. And this is the fact that injury to one group of sensory neurons can actually cause hyperactivity of uninjured bystander sensory neurons. So the cell bodies that happen to sit nearby those other neurons, they may not be directly injured, but they themselves can become hypersensitive after nerve injury that happens. And I'll show a little bit of the data that helps us to understand some of this. So nerve injury studies have shown this to be true. And ultimately, I'll bring up a couple of things that came out of a review that we put out, but ultimately where we're going with this is really trying to use these tools to try to better understand bladder pain mechanisms and urinary dysfunction. Maybe we can, through this, improve some of our diagnostic or therapeutic approaches. So what do we know about nerve injury and this idea that there are bystander neurons that can be affected? So a lot of what we know comes from the spared nerve injury model. This particular model is one in which there's a surgical damage that's done, exotomy to particular branches of the nerve, kind of mimicking traumatic nerve injury. And what we end up seeing is a very specific pattern of hypersensitivity in the foot that occurs in this region of the foot here that's innervated by the sural nerve. And the interesting thing about this is that we don't injure the sural nerve. We injure the other two branches of the sciatic nerve, but the sural nerve is left intact or spared, so to speak. Despite the fact that these are completely normal neurons, we have not injured them in any way, the part of the foot that's innervated by the sural nerve is the part that becomes hypersensitive. The regions innervated by the nerves that we cut, they lose sensation, but this sural area is innervated by uninjured nerves and shows hypersensitivity or mechanical allodynia, this exaggerated pain response to inputs to the skin. And we've been able to show that there are a fair number of different types of modalities of sensation that are affected in this particular type of model. That includes punctate, pinpoint, mechanical input. We've also done some work to be able to look at other modalities of sensation. As I mentioned, there are all these different types of sensory neurons that are responsible for different modalities of sensation. And so in our behavioral studies, you really do look at various different in vivo behavioral readouts to try to get a very good insight as to the cellular mechanisms, which sensory neurons are going to be actually affected in our models. In this nerve injury model, we also saw that there is not just pain in response to static input, which is mediated by certain subtypes of sensory neurons. We also saw pain that's mediated by dynamic input, which is mediated by an entirely different set of neurons. And we saw this in both the glabrous and the haired skin using kind of a brush assay where we're brushing the surface of the foot. Excuse me. So this was kind of the experimental design that we used to use. And the dynamic brush assay was something that we ourselves have had to kind of optimize to be able to use for this. It's not something that's used very robustly throughout the pain field. But suffice it to say, excuse me, I'm getting over a respiratory infection of sorts. So I apologize if my throat doesn't last long here. But essentially we were able to find, excuse me, that after nerve injury, we could see that there is not just a hypersensitivity to punctate inputs, but also this hypersensitivity to dynamic inputs that are moving across the surface of the skin. And that does implicate a very different subpopulation of neuron that might be involved in this hypersensitivity. We were also able to see that the changes that are happening in the haired skin, which has a different pattern of innervation, different types of neurons that innervate it, is very different from what we see in the glabrous skin. The two types of, excuse me, the nerve innervation is different, but we're seeing hypersensitivity in both of those modalities. So here's just kind of a general overview of the different types of assays that we've run, some of the different types of specific cell types that we think are involved in this. with nerve injury, we see a really wide, robust change that's happening kind of across all of these sensory modalities. But when we went and we actually looked a little bit more closely at some of our bladder pain models, we were actually able to see something that's a little bit different. So kind of keep this general idea in mind, and I'll go over some of the data that we've got from that in a moment. Other changes that have been reported on a more cellular or molecular level include changes that are happening in molecules within uninjured neurons. So we're seeing increases in some types of neurons that are usually associated with kind of our classic nociceptors. So CGRP is a very common molecule. TRPV1 is another common molecule. Both of these are typically only expressed in pain-associated neurons. They're not usually expressed throughout various different subtypes. But in our models, when we looked at pain, we could see that there were changes happening in uninjured neurons that included an increase in these particular types of markers, in addition to increases in spontaneous firing in certain types of subtypes of neurons as well. And this is somewhat summarized in this image that we've got. So there are a variety of different neurons that are present in the dorsal root ganglia. If you injure them, you can see changes in them that include this kind of trend of various different molecular markers. But you can also see changes that are happening in the uninjured neurons as well, that are kind of summarized here. Interestingly, sometimes it's actually the opposite direction of what is happening in injured neurons. But when you've got this mixture of neurons living together in the dorsal root ganglia, it's not just the neurons that are injured that are affected. And we found this to be a very intriguing phenomenon to be able to move forward with as we're starting to understand, okay, now what's happening in cases of injury that's happening in our viscera or in our bladder in particular. I will just kind of summarize here, just really reiterates the point, the take-home message that uninjured neurons can also contribute to hypersensitivity and can contribute to pain that's experienced in the hindpaw. So this is what we kind of are bringing with us to the picture, to these questions that we have about bladder pain. And the dorsal root ganglia is particularly interesting to us because there is a pretty good amount of just proximity between the hindpaw and the bladder. And one of the reasons why this is something that has really caught our attention is because it's pretty well known and well established that in mouse models of cystitis, for example, those mice exhibit hindpaw hypersensitivity. And if we look at just the basic anatomy of the neural anatomy, so to speak, of the bladder and the hindpaw, the innervation of the bladder is in the sensory innervation, has its root in dorsal root ganglia that are located very, very close and somewhat overlap with the DRGs that innervate the hindpaw. So, oh, I'm sorry, was there a question? Okay, there is at least a little bit of kind of an anatomic rationale or some evidence for this kind of potential anatomic links or anatomic proximity. And actually, I'll come back to this because we've been able to fill in some of the gaps here that actually help to strengthen the rationale for the potential interface between sensory afferents of the paw and sensory afferents of the bladder. And here are some questions then that come out of these kind of two bodies of work, or the things that we're bringing to this. Our big questions are, if there is injury, let's say to the hind paw, and those neurons are not the only ones affected, if there are uninjured neurons around those, could it be that some of those uninjured neurons that are affected are actually bladder afferents, or vice versa? If there is damage that's happening in the bladder and those sensory neurons are affected because of that damage or that disease, could that also be affecting uninjured paw afferents, for example? And so some of the evidence that we find, as I mentioned, mouse models of cystitis, there is hind paw punctate allodynia that has been very well established and published quite a while ago, and very reproducible. In addition, the more and more that we actually start to look for evidence of this potential crosstalk, the more and more we find. So female patients that have chronic pelvic pain actually show somatic mechanical sensitivity in areas of the skin. And in addition, those patients that have chronic pelvic pain are actually 100 times more likely to have a loss of what are called intraepidermal nerve fibers compared to kind of the populations at large. And so the thing that's really interesting about this is that intraepidermal nerve fibers, this loss of intraepidermal nerve fibers, this is diagnostic of a small fiber neuropathy. And essentially what this is it's looking at the sensory neurons that innervate the skin and actually seeing that there is a loss of the axon terminals in the skin. And so this is not the spinal cord that's affected. These are primary sensory neurons that are actually lost in the skin in a pattern that looks exactly like nerve injury pain. Interestingly, this pattern is seen in 100% of the patients that actually ended up having lower urinary tract signs or abnormal urodynamics in this particular study. And so this really kind of brings up the possibility of a couple of things. One, there might be somatic neuropathy underdiagnosed in patients with visceral pain because we simply haven't looked to see, okay, is there really a change in the way that they're sensing things or sensing stimuli in the skin? Are we looking at skin biopsies like they did in this study to be able to look at those nerve fibers? And finally, these are changes that are happening in primary sensory neurons in the peripheral nervous system, not the CNS, not the spinal cord. And another interesting thing that I've always found to be really intriguing, and I'm actually very excited about the talk that's coming up next month to hear a little bit more potentially about this. But another thing that is, an emerging therapy is that neurostimulation of nerves of the hind limb can actually alter bladder dysfunction in human patients. And so there really is a lot of kind of really interesting phenomena that really link hind limb innervation and the bladder. And we're interested in exploring this a little bit more so. So some of the questions then that we have, if we're looking a little bit more closely at this particular diagram, include questions like after spared nerve injury. So if we were to injure branches of the sciatic nerve, kind of indicated here by this X, which I hope that you all can see. After that happens, we know that there are changes in uninjured afferents, some of the uninjured neurons that might be here. Could it be that some of those neurons that are altered are actually bladder afferents and vice versa? And so this could be, instead of thinking of the spinal cord as the mechanism or the home of referred pain, it could be that interactions within the DRG or between DRGs could actually be a potential mechanism for referred pain. And this is a hypothesis that we're exploring right now using an array of different approaches. So to do this, we've started to look at both nerve injury and what happens to the bladder and bladder function, and also to look at bladder disease and what happens to hind paw afferents. So to understand bladder disease, we've adopted the intravesicular acrolein model of cystitis. And we've started to explore a little bit of that. That's a new model for the lab. So some of the things that we first had to do really were to get the model up and going, and then also to be able to look really very closely at some of the phenotypes that are happening, to be able to look at them with that fine-tooth comb, sort of those different modalities of sensation, different parts of the foot, and really dive in to understand more about what this particular phenotype looks like, and what that may tell us about the potential anatomic or cellular constituents of the pain that emerges after cystitis. So we have a assay in the lab that's looking at, it's called a void spot assay. So we're using that to be able to look at bladder, at urinary dysfunction, essentially, at voiding dysfunction. This has been something that's been that has been worked out by other labs, and that we've adopted with a lot of their input and a lot of hard work from our grad students. And so we're able to essentially look at patterns of voiding of a mouse on paper, and to be able to distinguish voiding dysfunction as a result. And so in our cystitis models, we've been able to kind of use this as a way of helping us to know that our cystitis is actually working, that we're getting our some sort of voiding dysfunction in our cystitis models, that we can then go in and kind of explore a little bit more closely. We have essentially looked at 2 different doses of acrolein to be able to study what's going on. With our lowest dose of acrolein, we don't see as much of a difference between kind of our baseline, which is here on the left, an initial time point that's about a day later, and then another time point that's 2 days later, about 48 hours out. On the top, I'm showing vehicle. On the bottom here is the low dose acrolein. And in our hands, we were not seeing a lot of changes in terms of bladder dysfunction or voiding dysfunction in particular at this particular dose. But if we used, a higher dose, and we have a lot of intermediate doses that have very similar results, what we're able to see is, of course, at baseline, all of these groups are comparable, but 24 hours later, we're seeing an increase in really small diameter voiding spots that really indicate to us that there is voiding dysfunction, kind of the inability to retain the urine and have urination in really large normal spots for a mouse, in a way that's very pathologic, and that is certainly characteristic of cystitis. So the interesting thing is that for both of these doses, when we looked at the hind paw behavior, we were actually able to see hypersensitivity in the paw, even at this low dose, but we're not seeing a very robust urinary phenotype. So we're interested in exploring these and kind of replicating this data a little bit more, but we may have actually found an interesting way to be able to look at cystitis, the effects that this has on sensory neurons and on the sensory nervous system in general, in a way that could be somewhat distinct from the urinary dysfunction. So there may be an involvement of the nerves here kind of above and beyond, or maybe before, at a lower dose, before we're actually seeing some of the actual urinary dysfunction. So that's an hypothesis that we're interested in pursuing and seeing if we can understand that a little bit more clearly. I will skip through some of these things. So some of the other types of questions that we have are actually looking at bladder afferents and using whole mount multiplex staining of the bladder to be able to explore innervation patterns and how these are changing. So we're able to do this in a couple of different ways. We certainly utilize immunostaining, but one of the things that's actually been a really great tool for us in understanding skin innervation and pain that's happening after nerve injury, we also have some tools that we've been able to use to look at very specific cell mechanisms. So looking not just at CGRP labeled nociceptors, but really to be able to look at other types of neurons as well. And this is really important, we think, for being able to study, for example, mechanosensitive subtypes that might, for example, be particularly activated by touching the foot or brushing the foot. So being able to study these would be really useful for the bladder, as these also would be the types of neurons that, for example, are activated by expansion of the bladder and may be differentially affected in our bladder diseases. So we have an array of different genetic tools that we've used in the skin in the past to study mechanosensitive neurons that we're now starting to use. And we found that at least 2 of those mouse lines are going to be very useful for studying bladder innervation as well. This is an example of just kind of one of them, but we're able to get a little bit of a sense of what that bladder innervation looks like, being able to quantify that bladder innervation in different regions, including the neck, kind of the body, and then the dome, which is kind of shown here. And we're able to do this using one of our microscopes that we have available, to do this in whole bladder, so that we're able to look at this with kind of a very nice distribution of innervation throughout the tissue sample. One of the things that I mentioned that we wanted to really do is to take some of the tools that we have and to explore the phenotype that we're seeing in these animals after nerve injury. And one of the things that we found to be really interesting is if we actually look at the paw after cystitis, what we were able to see is a very spatially restricted region of hypersensitivity that is specifically in the sural area. So very similar to what we see after our SNI animals, as a matter of fact. If we probe the middle of the paw, we're really not seeing any difference between the baseline and the animals after acrolein. But we are certainly seeing a hypersensitivity, and this is something that we've been able to reproduce several different people in the lab. So we really think that this is kind of bona fide and very intriguing data, that we're really seeing hypersensitivity very specifically in the sural region. And so it's very interesting to see this because it very much parallels what we were seeing after our nerve injury. And it also really implicates for us a potential role for sensory neurons that are coming specifically from the L5 dorsal root ganglia. And one of the reasons for this is because that is actually the DRGs, kind of one of the key DRGs that are very, very interesting for studying what happens to uninjured neurons in our spared nerve injury, where we're also seeing hypersensitivity in that same exact area, the kind of red dermatome, so to speak, that's delineated here in this kind of cartoon. The other thing that's interesting about the L5 dorsal root ganglion is that there are some reports that there may actually be bladder afferents in that ganglion. So if you are looking at the literature, or if your attempt is to try to pick a dorsal root ganglia that has the most bladder afferents possible, often in mouse studies, L5 might not be actually mentioned. But what we really wanted to do is ask, is there anatomic evidence for a DRG that houses a bladder afferent and that houses a hind paw afferent, particularly the afferents that innervate this specific part of the foot? And we've been able to find that, yes, in fact, there are. So we did some studies using 2 different types of tracers. One tracer injected into the foot, targeting that hypersensitive area, and another tracer that was introduced into the wall of the bladder. And we can see, as you can see some evidence here, We've got bladder afferents that are shown in green and hindpaw afferents that are shown in blue that are showing up in the same dorsal root ganglia. And so this really does provide us with at least some of the a potential anatomic substrate, essentially, for the potential for intercommunication between these different types of neurons. And we found that to be really quite interesting. So ultimately, what that means is our little sketch that we were looking at earlier really looks a little more like this, where in our L5 DRG, there are a few bladder afferents that are there. When we did the study, we looked at a really wide variety of dorsal root ganglia. And so went up, I think, as far as the more caudal thoracic levels, kind of all the way back through S1. And we were also able to see that there are a few individual paw afferents that you can actually see up in some of these more rostral DRGs as well. So ultimately, we do have the kind of anatomic substrates for a potential crosstalk here that's going on. And that's really interesting for us to be able to look at. And it gives us a really good, a good place to target if we want to go in and look at some of the molecular changes or the neurophysiologic changes. We can go in and actually target these specific types of dorsal root ganglia to be able to ask deeper questions. The other thing that this really brings up for us then is in our approaches, we really want to study intact dorsal root ganglia because we really want these kind of local circuits to really be present. Dorsal root ganglia are kind of phenomenal for being able to dissociate them and to do cultures. They are relatively hardy, particularly the smaller diameter neurons are relatively hardy, but being able to keep those intact is something that we've really tried to prioritize in our approaches. And that's something that, excuse me, for us has meant that we also need to be able to kind of develop new ways of being able to look at those and refine new ways to be able to look at those targets or look at those intact circuits. So one approach that we've used is to develop an ex vivo skin ganglion cord preparation. And essentially what we're able to do is to take kind of this diagram here. So we've got the foot, we've got the nerve, and it connects to the spinal cord and the dorsal root ganglion. So we're able to recreate this. So we've got skin from the foot, the nerve, and the spinal cord with little itty bitty DRGs that are attached that are kind of delineated here. And so we can actually then go in and study what's happening to those dorsal root ganglia neurons without having to cut the neurons first. And so we think that this preparation is going to be something that will be really quite a powerful way of being able to address some types of questions about the ways in which these neurons are interacting with each other within their dorsal root ganglion, and to kind of see what happens if there are, for example, hindpaw afferents that we can actually stimulate in a very naturalistic way by poking or stroking the skin. Are those neurons actually more active, or are we seeing a difference in the signals that are being recorded here if this is a preparation coming from an animal that's been through cystitis? We're also interested, as I mentioned, in labeling very specific mechanosensitive subtypes. And so being able to kind of include our genetic strategies to label different types of sensory neurons, I think is going to be very powerful for us in understanding a little bit more about the cellular mechanisms of bladder pain. And so this strategy that I alluded to before, we were able to see a pretty good number of CGRP-negative bladder afferents using some of the exact same genetic strategies to specifically label different sensory subtypes that are good for labeling mechanosensitive afferents in the skin. So using both TrkB and also RET-based promoters to be able to drive KURe expression, and then using KURe-dependent reporters, we've been able to generate This is a kind of a 3D stack of a region of bladder innervation, I think from the body of the bladder, if I'm not mistaken. So you can see here, we've got our labeled potentially mechanosensitive afferents that are really quite distinct from the CGRP that's here. And then we also have some degree of neurofilament heavy, which we've used to be able to delineate kind of a less specific marker of highly myelinated neurons, afferents. And here are kind of two examples, and just kind of comparing a little bit of what we're able to see with some degree of overlap with CGRP, but indeed there is certainly some afferents that are not dual labeled that are present, and that we think will mean that this tool will be very interesting for being able to study what's happening to those afferents in our models. So with that, I will stop and send a very large number of thanks out to the Crawford crew, to the team of folks that I have working in the lab, including graduate students, veterinary students, lab technicians, research interns, and undergrad students that have all lent a really helpful hand in a lot of different parts of the projects that we participate in, as well as my mentors on the K-12 grant that I have, both Dale Burling and John Sfarin, and various different collaborators that are here at UW, including Chad Vizina's lab and Kim Kilstites, as well as some of the collaborators that I also have that have been really instrumental in helping us to understand the genetic tools that we have for being able to study nerve injury pain and really kind of translate that over into studying bladder pain. So that includes Michael Caterina, as well as the lab of David Kinty.